Okay, it looks like the attendees have filed in, so we'll begin today's webinar, which is the use of plant-derived antifungal Poacic acid for the use in turfgrass disease control. My name is Laura Isles. I'm one of the co-directors of the North Central IPM Center. We're one of 4 regional IPM centers supported by USDA NIFA Crop Protection Pest Management Regional Coordination Program. And we're on a mission to coordinate IPM across the region by sharing and promoting IPM research, extension, and teaching activities. In a minute, we'll start, but if you have any questions during it, there is a question and answer part at the bottom of your Zoom, and please use that to answer any questions, post questions anytime, and then we'll answer the questions after. So today's speaker, Paul Koch from the University of Wisconsin-Madison, will talk to us about antifungal Poacic acid. I've been practicing that pronunciation. I'm probably still a little bit wrong, but I'll let you take it away, Paul.
All right. Thank you for the introduction, Laura, and thank you to the North Central IPM for organizing and coordinating and moderating the relatively brief webinar today. And of course, thank you to the USDA Crop Protection and Pest Management Program for funding the work that I'm going to present to you all today. And thanks to you for tuning in to learn a little bit more about this particular compound and the work that we're doing. So what I'm going to do today is give a brief update on a project related to Poacic acid. And so I'll talk about what Poacic acid is, what we're doing with it, and where we're going with this particular product. And the name of the project is for use in turfgrass disease control. When I talk about turfgrass disease control today, I'm really talking about dollar spot. So dollar spot is the most common disease of turfgrass in temperate climates, golf course turfgrass, in temperate climates around the world. Certainly in the Midwest, where I'm based in Wisconsin, it's our most important turfgrass disease, commands the vast majority of fungicide applications. In certain instances, we can apply 8, 10, maybe even more fungicide applications per year, targeting just this particular disease. If left untreated, oftentimes it can cause serious damage to the turfgrass, to the golf course, and make it virtually unplayable. Dollar spot is active at a very wide temperature range, and so it's active for a very large portion of the year. Really, in the Midwest, it can be active from mid-May until into early November in certain years. If we look at other parts of the world, in particular Europe and Scandinavia, we're seeing dollar spot increase in severity due to climate change. Dollar spot traditionally has not been a severe disease throughout many parts of Europe and Scandinavia, but just in the past 5 or so years, they're seeing the severity increase significantly with the changing climate, and they don't have the large number of chemical options throughout Europe and to a slightly lesser degree in Canada that we have here in the States. And even here in the States, what we're noticing is that increasing cost of fungicides, rapidly developing fungicide resistance, and greater concern over human health and environmental concerns are also limiting the options that we have for chemical control here in the United States. There's been proposals from the EPA to limit or remove multiple products that we use commonly for dollar spot control. And so that'll make things more difficult moving forward. So really the gist of this project is to try and find another option that we can use and still get acceptable or effective dollar spot control. And the other option that we're testing here is a plant-based product called Poacic acid. So what is Poacic acid? Well, this is the paper that describes the discovery of the compound. So if you really want to learn more about the compound itself, I would recommend going and reading this paper, Piotrowski et al. A group of researchers, many of them based at UW-Madison within the Great Lakes Bioenergy Research Center. And what they were doing as part of the research was screening a bunch of compounds for antifungal activity. Many of these compounds were were found in the lignocellulosic hydrolysates of grasses, basically byproducts of biofuel production. So that, as they're using biofuels to create energy, it's producing a lot of waste. And so what this group was doing was screening that waste for various compounds and their antifungal activity, especially for use as antifungals in plant disease control, right? So if you can kind of isolate some of these compounds from plants, maybe some of them are related to defense compounds for against plant disease, and can be used as sort of a plant-based fungicide. And that's exactly what this is. One of the more effective ones that they isolated and found was Poacic acid. And the chemical structure is pictured here. And also the in vitro activity on soybean plants against Sclerotinia sclerotiorum was found to be pretty successful. However, when they first tested this in the field on soybeans, they didn't find a lot of success. And one reason that they thought they didn't see a lot of success is the time interval, the reapplication interval is relatively short. The time this compound is active is relatively short. And when you have hundreds of acres of soybeans to spray, you can't go out there every couple of weeks and reapply the antifungal agent. In turfgrass, we can. So after really not seeing a lot of success in soybeans, Mehdi Kabbage, my colleague here in the Department of Plant Pathology at UW-Madison, came to me and said, Paul, why don't we test this on turf? And so we did. And so the paper that details those projects is this paper that we published back in 2020 in the journal Plant Pathology. And so we basically tested Poacic acid, the raw Poacic acid compound, against dollar spot and snow mold. And what we found is that in vitro, very, very effective, excellent suppressant of both dollar spot and snow mold. So the figure that we just brought up on the right side of this figure, the top cultures and the top bar graphs are dollar spot, and then the bottom cultures and the bottom bar graphs are snow mold, pink snow mold in particular. And in vitro, excellent suppression, anywhere between 200 and 500 milligrams per liter as far as the concentration that proved effective. We went then and tested this raw Poacic acid product mixed with DMSO and water in the field. And what we found is that at these concentrations that we tested at, they were kind of effective. They weren't great. They weren't commercially acceptable. We tried mixing them a little bit with fungicides and really didn't find any added benefit. And so what we were thinking and what we noticed about the product is that the raw Poacic acid product is extremely hydrophobic, really, so it does not mix well with water. And, we know with talking with some of the larger chemical companies, they put huge amounts of resources into formulation. And here we are working with this completely raw plant extract, basically, and that's very hydrophobic. And taking the next step, where do we want to go? We wanted to try and see if we could partner with somebody to formulate Poacic acid or come up with a formulation that would reduce some of the hydrophobicity, make it reside on the plant, spread out over the plant more effectively, and hopefully provide more effective plant disease control. And so that was the basis for our grant to USDA CPPM, which was titled Incorporating the Plant-Based Antifungal Agent Poacic Acid into an Integrated Disease Management Program in Turfgrass. 3 objectives with this project. Objective 1 was to partner with private industry to develop and identify 1 or more effective formulations for Poacic acid. Objective 2 then was to look at how those formulations could be integrated into a turfgrass disease management program. And then objective 3 was to disseminate that information out to turfgrass managers in innovative ways. So I'm going to mostly talk about objective 1 and 2 over the next few minutes, give you an update on where we're at with those and then where we're going over the rest of the project. So we actually partnered with Simplot Turf and Horticulture. They're a company that we have worked with at UW in the past. They're a company that we know has done work in sort of alternative type products and fungicide alternatives. So we talked to them about coming up with a formulation, a formulative that we could add to the product. And they worked with a subcontractor who came up with an adjuvant that, as you can see in this picture, dramatically reduced the hydrophobicity of the mixture and allowed it to spread out over a waxy paper. But certainly, if applied on a plant, we would anticipate that it would also spread out over the waxy leaf cuticle on a turfgrass leaf blade as well. So objective 1, we've come up with a formulation that reduces the hydrophobicity. Now we move on to objective 2, and we look at, does the formulated product improve disease control compared to the unformulated product? Well, so in objective 2, we had 3 different factors that we tested. One, we tested 2 different rates of Poacic acid, 0.97 fluid ounces and 1.94 fluid ounces per 1,000 square feet. These are the same concentrations that were tested in our original plant pathology paper, 500 milligrams per liter and 1,000 milligrams per liter. Then we did it with and without adjuvant. We didn't know the impact the adjuvant would have. So when we did test with adjuvant, we actually tested at 3 different concentrations. That's 5, 10, and 20% volume by volume. And then we tested different reapplication intervals, 7 versus 14 versus 21 days. In a turfgrass environment, 7 days is really short. It's really difficult, but on your putting greens, you could probably manage that. Many golf courses do go out and spray something on their putting greens every 7 days. When you get to the larger fairway acreages, 7 days becomes more impractical, more difficult. So we wanted to see if we could get any efficacy out of those 14-day and 21-day reapplication intervals and make it more applicable to a wider range of uses on the golf course. So I'm going to show you some results here. We haven't done a lot of statistical analysis on these results. These are all from last summer, 2022. On the x-axis is a few targeted treatments that I brought out. We have 26 overall treatments in this particular objective 2 project, but I'm not going to go through all 26 of those treatments. On the y-axis, we have dollar spot AUDPC, which stands for area under the disease progress curve, which is basically just a summation of all the dollar spot that occurred over all of the rating dates. So we rated dollar spot every 2 weeks throughout the growing season from mid-May until early November. And AUDPC is just kind of a single summation of all those ratings for each one of these treatments. What we noticed is that the lower rate of Poacic acid really had very little control against compared to the non-treated control, whereas the higher rate of Poacic acid did reduce it nominally, not a lot. This was I don't have error bars here, but this was a statistically significant reduction compared to the non-treated controls. The higher rate of Poacic acid is showing effects compared to the lower rate of Poacic acid, but nowhere near the level of control that we would need for commercial viability, right? So you see the fungicide program there, the furthest bar on the right, that program is performing far and away the best. So just the Poacic acid at that high rate is not performing where we need it to perform. And here's some pictures to kind of show you the degree of the disease. We didn't have a tremendous amount of disease pressure actually on the plots last year for whatever reason, but so there's the DMSO control and then the Poacic acid at the lower rate, which here is this 1250 mils per acre, and not really any reduction. We are seeing clearly a reduction at that higher rate at the 7-day reapplication interval, but this is still would be considered unacceptable in most golf course situations. All right, so what about the impact of the adjuvant? Again, I've just kind of pulled out some select treatments to give you a feel for the impact that the adjuvant is having when combined with the Poacic acid. All of these are the high rate, the 1.94. So I told you before, the lower rate really didn't have much of an impact. So I just focused on that higher rate. And what we were seeing with that higher rate, we did see a significant reduction at the 5 and the 10%, but then we saw a further significant reduction at the 20% adjuvant when mixed with the high rate of Poacic acid. Again, what we see is that the fungicide is far and away much more effective in this particular project, and we're not getting a commercially viable level of dollar spot control, even when we have the higher rate of Poacic acid and the highest concentration of adjuvant. So we're seeing some success, but not the amount that we would need to show that this is a commercially viable option for disease control in turfgrass. But what kind of has given us optimism for moving forward is that the single best treatment of all those 26 treatments, it was the highest rate of the product at the highest concentration of adjuvant at the shortest reapplication intervals. We're clearly seeing the large amount of reduction in that particular treatment, not quite to the fungicide program, but also not terribly far off. And so when we think about what we can get out of this first year of the study, is that we're seeing some success here, but maybe we're just not at the rates that we need to. And if we think back to what we learned from the original paper that we published in Plant Pathology back in 2020, if I could have done that first year over again, we would have gone at higher rates. Because even with a synthetic fungicide, if you test a particular concentration that's effective in vitro in the laboratory, you always need to increase that by a factor of 5x or 10x once you go out into the field, because of all the different environmental factors that are impacting that fungicide once it goes out into the field. So that's kind of where we're going to focus some of our future work. So we're going to increase the rates of Poacic acid to basically a 5x and a 10x rate compared to that 1.94. We are also going to conduct a demonstration on the University of Wisconsin's University Ridge Golf Course to show its efficacy in a real-world golf course situation. Next year, we hope to hold a virtual field day to discuss the results of this product with a global audience, probably do a Zoom type field day. And we had a setback with this particular year. We used all the Poacic acid that we had on hand last summer 2022. So we had worked with Simplot to find another producer to make more Poacic acid, because it was just going to be prohibitively expensive for us at UW to make more. And we were supposed to have the product in mid-April. It was all going to work great. And then we just had delay after delay after delay. And then actually last week, we just kind of pulled the plug and said, you don't have it to us yet. You've been telling us it's going to be there next week for the past 2 months, and we still don't have it. So we're late enough into the summer now that we wouldn't gather a whole lot of data. So we're just going to postpone, hopefully find a new subcontractor to make some more Poacic acid for next year and request a no-cost extension on the grant and do this year two's worth of work in 2024. Hopefully these new rates will give us some better disease control, show it as a commercially viable product, either on its own or when mixed with other products, and hopefully set it on a path towards being a useful product for plant disease control in the future. So with that's all the information that I have. If any of you have any questions, please feel free to call me up or email me. We do have the full annual report at our website, tdl.wis.edu/results. So if you just kind of look through those reports, look at the Poacic acid one, you can see the full report for year one. And a huge thank you to Simplot Turf and Horticulture for doing a lot of work with us on producing the adjuvant and working with us to get this batch of Poacic acid produced. And of course, thank you to the USDA CPPM again for funding this work. So with that, I'll stop there and I'll answer any questions that may have come in.
Thank you, Paul. That was great. So first question that came in is, hi, Paul, will you be testing against microdocium? Well, this is like the hard, I'm an entomologist. Microdocium.
Oh gosh, well, you should see us trying to pronounce insect names. So I see it's from John. I know John overseas, and we will eventually. Certainly, microdocium patch is a severe disease in the UK and Northern Europe and Scandinavia. So we're doing the first round of work on dollar spot. We will be doing a bunch of in vitro testing against a range of turfgrass diseases. And certainly microdocium patch is another one. That'll probably be second on the list that we test against.
Okay. Yeah, please, anyone type in any questions. I just had, you're looking to increase the rate. I mean, do you think at the point will it become cost prohibitive? Do you expect any phytotoxic issues? Like, kind of, I guess, how far can you push it? We
haven't seen any evidence of phytotoxicity yet. So I'm optimistic that we will not see phytotoxicity, but that would certainly be one of the things that we look at closely as we increase those rates. As far as the cost, I have no idea once we go to a commercial scale, what it would cost to produce the product in a commercial scale. So that one's a tough one for me to answer.
Right.
To get us about on a small scale, on a contract scale, it costs about $25,000 to get 500 grams. So that's a lot, but once a company would work on a commercial scale, I'm sure those costs would come down dramatically.
Right. Let's see. And you're working on other plant diseases. So is it mostly for turfgrass diseases? Are there other plant diseases that seems to suppress?
Yeah, well, I don't anticipate that this product will have a very long residual. So I don't think it's going to be active for like a lot of synthetic fungicides that are on the market today will have 21, 28 day or more residuals where they're active. I don't think this product will have that. So will it be used in row crop agriculture where you can only make one application and will need a fairly long period of time for it to work? I don't know. I think its market might be relatively limited there, but smaller, higher value type of systems like, like apples or strawberries or even potatoes. Potatoes are sprayed on a very regular basis oftentimes. So in those kind of cropping systems where it's not time or cost prohibitive to go out every couple of weeks, I think this product certainly would have potential usefulness there.
We had another question come in kind of, I guess, on the mechanism of how it works. It seems to directly affect the pathogen. Any effects on plant defense response, do you think?
Yeah, it's another good question from John. I don't know. We haven't tested that. I would be surprised if there is no impact on the plant defense because it is a byproduct of the biofuels process from grasses, right? So it's basically like almost like a host grass chemical. So I would be surprised if there was no plant defense response. We haven't tested for any plant defense response. We haven't seen any evidence of plant defense response, but I'd be surprised if there was no plant defense response at all.
Very interesting. Do you think any risk of resistance to this, or how will this compare to kind of fungicide and resistance problems?
Yeah, I mean, certainly, I think we would want it to be used as part of in a plant or in a fungicide resistance program right away. It targets the cell wall development structure, the beta-1,3 glucans in fungal cell walls, appears to bind at a single site. So we know that's a recipe for resistance to develop. So right from the start, we would not recommend that you're only applying this product over and over again throughout the course of growing season, but that you would take mix, rotate with other types of things that work on other areas of the fungus to mitigate fungicide resistance right from the start. There's been no documented evidence of resistance, but just based on how it works, I would be pretty surprised if there was no resistance that would develop.
Okay, so part of the toolkit, not something to replace.
Correct.
Okay. Any other questions coming in? Okay, we got some thank you from there in the chat, if you can see the chat there. So no, thank you so much. This was really interesting and enjoyed learning about it. So and looking forward to, I guess, when you get the more results from 2024 and on, it would be great to do a follow-up on this. So
Definitely, we're looking forward to it.
I really appreciate it. So as we wrap up our webinar, our next Pests and Progress webinar will be on Wednesday, September 20th. We're skipping August, and on September 20th, again, it will be at 12:30 p.m. Central Time, 1:30 Eastern, and we're going to talk about certified crop advisors' perspectives on pesticide resistance management. So that should be an interesting one, continuing our resistance management discussions. So we look forward to seeing everyone in a couple of months. And thank you again so much, Paul.
Thank you.